Welcome to the Cancer Genome Atlas database, also known as TCGA. This comprehensive database houses an impressive collection of over 20,000 primary cancer samples spanning across 33 different cancer types. This vast repository represents one of the largest cancer genomics databases available to researchers worldwide. TCGA is a treasure trove of multidimensional data. It encompasses genomic information revealing DNA alterations, transcriptomic data showing gene expression patterns, epigenomic data highlighting DNA modifications, and comprehensive clinical data tracking patient outcomes. This multi-layered approach provides a complete picture of cancer biology. The power of TCGA lies in its ability to unveil cancer-causing mutations through comprehensive genomic analysis. Researchers can trace the intricate mechanisms of tumor development, leading to important discoveries. This valuable resource has been instrumental in identifying and validating new therapeutic targets, advancing our fight against cancer. Every piece of data in TCGA undergoes rigorous quality control processes, ensuring researchers have access to reliable, high-quality information for their studies. TCGA's applications in modern cancer research are many. Machine learning models developed using this data can predict cancer outcomes with increasing accuracy. The database facilitates the discovery of biomarkers for early detection, potentially saving countless lives. Researchers can predict drug responses, personalizing treatment approaches for better patient outcomes. Additionally, TCGA helps us understand tumor heterogeneity, revealing why some cancers resist treatment while others respond favorably. This knowledge is crucial for developing more effective therapeutic strategies.
So let's start exploring the TCGA data portal. At the top, we can see we have Analysis Center, Projects, Cohort Builder, and Repository. We do not know what sort of data set we want to get from GDC data portal. So first we have to get the data set using Cohort Builder. So let's dive into the Cohort Builder. Here we need to pay attention to these attributes. So these are the attributes from where we can filter our required data set. Let's see if we get the open access data. Yeah. Under the access section, we have controlled and open. So we need to select the open data since controlled data can be difficult to access. Yeah, so we have open data. Now, since we want to perform biomarker discovery, we need gene expression data, so which we can find under data type. I will select gene expression quantification. So now we can see all of the other options are now restricted. And then obviously we need RNA sequence data given by experimental strategy. Since we want to do biomarker discovery with our TCGA data, we'll go to Biospecimens tab. Then we will select the tumor type, which is given under tumor descriptor, mainly the primary. So this was our main criteria for getting the cell affected with tumor. If we want to dig down further, then we can use additional tools such as alcohol history, tobacco smoking, number of cigarettes per day, or what kind of treatment they are taking currently, or what treatment they had earlier. Similarly, whether they had some other diseases or which stage of cancer they are currently. So all of these things, if we want to dive into further details, then we can use these attributes. Now we have come to the general section. As you can see, we have 19,585 cases. These are the cases which comprises from different projects. So we are only interested in finding out the breast cancer. So the appropriate project for breast cancer would be breast invasive carcinoma, which is the acronym TCGA-BRCA. So we have total of 1,095 cases. So we will be selecting this project. Now we have 1,095 cases, and we can see the filter applied using the cohort builder. Now we are interested in downloading the dataset. To download the dataset, we have to fetch it from the repository. So let's close this code builder, go to the repository section. Okay, so we have landed into the repository section where we had applied previous filter using code. Now we have to download these files, but as you can see here, interesting thing, we have 1111 files with 1095 cases, and the file size is 4.71 GB, which is really huge. So downloading this data manually takes a huge time, so we have to use a special tool. We need to download a special file called a manifest file under this section. After downloading the manifest file, we have to add all of these files to the cart so that we can also get access to the clinical data associated with these files. Now go to the cart section at the top. To download the clinical data, we have to click on download associated data, then select clinical, either TSP format or JSON format, whatever we want to process. So I'll take TSP format. To download the large files, 4 point something GB, we have to use the special GDC data transfer tool. So here is the download page. So I have already downloaded the tool, and then we need manifest file, which was downloaded previously. So I'm going to add this manifest file. So it will do its job automatically. The dataset has been downloaded, and we have clinical data, and we have the expression data for these files, which we downloaded using the manifest. Now let's explore our gene expression data. Here we have rows and columns. Each row corresponds to a unique gene identified by its ensemble ID. The numerical values represent gene expression levels. Similarly, in the columns, the first column contains ensemble ID for each genes. Remaining columns represent samples identified by TCGA barcodes. Clinical data, we also have rows and columns. In rows, it represents individual patients or cases, which is identified by a unique submitter ID. In the columns, we have clinical attributes describing each patient's clinical history or medical status. Additional pharmaceutical therapy, additional radiation therapy, additional surgery, local regional procedure, age at initial pathologic diagnosis, etc. are the columns. Our analysis of the Cancer Genome Atlas dataset, breast cancer dataset, revealed important patterns across 1,184 patients. This scatter plot shows how patients cluster by cancer stage, with each dot representing a patient. Similarly, this graph shows our top 5 biomarker genes and how their activity changes across stages. This heat map reveals gene expression patterns across different cancer stages, where blue shows low activity and red shows high activity. Therefore, we have identified 20 significant biomarker genes that could help better understand breast cancer progression and potentially improve diagnosis.
The dataset excels in findability through its well-structured GDC data portal. This portal is designed to make it easy for researchers to locate specific cancer genomic data, providing a user-friendly interface and powerful search capabilities. Researchers can quickly find the data they need thanks to the portal's intuitive design and comprehensive metadata. This ensures that valuable time is not wasted in the search process. In terms of accessibility, TCGA implements a controlled access system. This system ensures that sensitive data is protected while still being available to authorized researchers. By balancing data sharing with privacy concerns, TCGA maintains the integrity and confidentiality of the data. This controlled access approach allows researchers to access the data they need without compromising patient privacy. It also facilitates secure data sharing among the scientific community, promoting collaboration and innovation. The interoperability of TCGA is evident in its standardized data formats. These formats ensure that the data can be easily integrated with other datasets and tools, enhancing its utility for various research purposes. By adhering to common standards, TCGA data can be seamlessly used in different computational environments.
TCGA data can be used many times by different researchers because it has clear instructions and rules on how to use it. This helps scientists build on each other's work and make new findings. In summary, FAIR principle. Rating, F findability, 95%. A accessibility, 90%. I interoperability, 95%. R reusability, 90%.